Morning. My name is Sean Mizo, and first I want to thank you all for joining me in person or virtually for my presentation. So my research topic is on detecting native Hawaiian freshwater fish using eDNA. My project began with Dr. Chan introducing me to this Iolani-based organization, Navai Ekolu. And so Navai Ekolu worked with countless volunteers from schools such as Palolo Elementary, Kamehameha Schools, and Kalani High School. And what they do is they try to improve the biodiversity within Oahu freshwater streams. Throughout their years of surveying streams, they found that there's about a ratio of 40,000 invasive organisms for every one native organism, and thus you can see the importance of their work. So Navai Ekolu surveys freshwater streams using pae nets, which are large A-frame nets. And so they go out into the streams, capture fish, identify them, count them, and then release the natives or remove the invasives. However, pae nets aren't perfect, and so I wanted to see if there was an alternative method that could be used in addition. After doing some research, I learned about environmental DNA, or eDNA. And eDNA is a relatively new method of species monitoring that's only been used by researchers for the past 10 years or so. So what is eDNA? In general, all species and organisms will release DNA into the environment. And as you can see on the right, taking one water sample of a lake or stream, you get DNA from snails, fish, algae, lizards, bacteria, or whatever lives in the stream. And in particular, fish will release DNA via skin, scales, feces, urine, or reproductive materials. And researchers have found that, as you can see on the left, that you can collect this DNA, extract it, condense it, amplify it, sequence it, and be able to tell, okay, this, and this species were in the water just from a water sample. Pretty cool, right? However, this method isn't perfect as well. And so I want to look at the pros and cons of both the pae and eDNA method. So for pae, it's relatively simple. You go out into the streams, capture your fish, count it, and on that day, you get the results. However, there's some drawbacks where you need lots of time, volunteers, and education in order to teach volunteers how to correctly identify the species of fish. And this comes with identification difficulties. Young organisms that haven't developed their identifying qualities yet are very difficult or near impossible to distinguish between one another. And secondly, the pae method has its own sampling bias, where by going out into the stream and splashing and using big pae nets, it only captures fish and species that can be caught with that method. However, eDNA, instead of having a bunch of volunteers come to the stream, you can have just one person go to the stream and take a water sample. Furthermore, this allows, since eDNA flows downstream, this allows for the monitoring of species without the entire stream. So for example, in places high in the mountains where it's difficult to bring a big group of people to a sample, you can just take one water sample at the bottom of the stream. However, this has its drawbacks as well. DNA is subject to degradation from many sources, and on a different day or different time, even if the number of species is the same, you can have varying DNA concentration amounts. Secondly, because it's so new, the procedures for using eDNA is on on-site, on-location basis, and thus using one in Hawaii, I'd likely have to create my own procedures to figure out how to best optimize for the environment. And lastly, environmental DNA also has a sampling bias where species that have low biomass or in low density population are likely to give less DNA and won't be able to be detected. So with these in mind, I created my research question of, can eDNA or environmental DNA be a reliable and feasible strategy to monitor freshwater species in a high school environment? And from there, I moved on to creating my methods. So the first step of any eDNA method is to collect your DNA. So there's two variables I first introduced here, centrifuge and sterivix. So centrifuge variable includes using a 50 milliliter tube of water to collect the water, and then come back to the lab and use a centrifuge to spin it down. This, in theory, will leave the DNA just at the bottom, so I can pipette just the DNA out. Next is the sterivix. So sterivix is the white part on the hand pump, and that's just a filter. So instead of getting a whole vial, I can push water through the filter, leaving the DNA just on the filter. Both methods have shown success in previous articles, however, I wanted to see which was best for Hawaii. Next is the DNA extraction stage. And this stage basically is trying to get the 50 milliliters of solution down to a few microliters of just DNA in solution. I used a Keogen DNeasy blood and tissue kit, which is simply a collection of buffers and enzymes to help this process. And on the left, you can see a more specific example, but the idea is that you have your DNA and try to get it down to just a little bit, as little concentration as possible. Sorry, as high concentration as possible. And however, this, you still have all the DNA, bacteria, fish, lizards, but we want just fish DNA. So going forward, this is the polymerase chain reaction step. So here, you can use specific primers to just bind fish DNA. So how PCR works, a quick breakdown, is you put the collected DNA in a solution with nucleotides, DNA primer, and replication enzymes. And the primer, which in this case we use the MyFish primer, will only bind to fish DNA. And when it binds, that DNA will be amplified, and this increases exponentially. So in theory, at the end, you only have fish DNA amplified. And as you can see on the right in the fine print, there's specific temperatures that are needed and time needed. This is done by the thermocycler. The thermocycler just is where the PCR occurs, and it holds the solution at specific temperatures at specific times. Following that, next is the DNA sequencing step. So first, a library prep stage occurred, where we prep the DNA solution to be ready to be sequenced. So the small rectangle is the actual sequencer. That's the Oxford ion. And that box is where we add the DNA and where sequencing occurs. So as you can see on the video, how it works is DNA is fed through a protein, and that protein will actually change shape depending on which base it gets. This protein shape correlates to an electronic signal that can then be read by the ion and then will be sent to the computer. So after the DNA is fed to the protein, the computer will be able to have a long list of all the base pairs and all the base sequences that it received. And so now I'm left with huge files of just a bunch of letters and base pairs that normally don't mean much to the average person. And so this is where these two softwares and databases come in handy. First is NCBI, GenBank, and next is Genius Prime. So GenBank is an online database that has over 1.6 billion sequences and which equates to 450,000 species. And this is just accumulated from past researchers uploading their sequences to the database. And what I can do from there is copy and paste my sequence and blast it, which basically just means to match my sequence to the database, and it'll come back on which species it related with. So in this example, as you can see, my DNA strand matched with Ancestris temaniki, which is a fish that was in the stream. And so Genius Prime is a software that can do this on an automatic basis. Instead of me copy and pasting every one, it does all of them for me automatically. However, something to note here is that the DNA sequencer I used previously, the MinION, although it is the cheapest on the market, can do long base pair readings, and it's the only one that can use on site, it is also the one that's prone to the most errors. So I tried to accommodate that with Genius Prime. First, I only counted DNA reads that came back as 70% or more identical. So as you can see here, this one is 100% identical, but if it was less than 70%, I wouldn't count it. And secondly, I organized the reads by genera instead of species specific. So with all these in mind, I set out to Carnegie Park to do my first sampling. I chose Carnegie Park because that's where Novaicolo did their sampling on the same day, so I can compare my data. And here are the results. So on the electrophoresis, the red lines show amplification, and on the far right lane, that is the latter. So as you can see, only centrifuge show amplification. However, more notably, as you can see in the pie chart, 98% of my reads were non-fish DNA. So something was clearly wrong, and I'd have to go back later to figure out what the errors were and how to correct for that. And so here are the genera found. The numbers underneath Novaicolo equate to the number of organisms that they found on that day, and the numbers underneath the 2 and 1 equate to the number of reads that I found using eDNA. And the yellow bars mean things that overlap. And off the bat, you can see not too many yellow bars and not too many reads. However, this was only the first sample, and I learned a lot from this one and able to go into the second sample with more knowledge. So this one occurred at Manoa Field, a little upstream of Carnegie Park, and once again, I selected this because that's where Novaicolo sampled on that day. The first adjustment was made to the PCR stage, and so as you remember the thermocycling conditions, I adjusted the temperatures and times to be more optimized for the fish primer. And secondly were the ampere beads. So these beads took place in the library prep stage, that was during the DNA sequencing step, and the ones that I used in sample 1 were a little expired, and so I used new ones in sample 2, and that resulted in much more better DNA yield. And here are the results from sample 2. So initially you can see from the electrophoresis that centrifuge and stereovics showed amplification, which is interesting since stereovics showed no amplification in the beginning. However, more notably in the sequencing results, instead of the 98% non-fish DNA, only 45% non-fish DNA. So the changes were much better and showed much improvement. And here's a table of the genera they found on the day. Off the bat, you can see much more yellow and also much more reads. Here, the reads range in the thousands to 10,000s, whereas before, some didn't even reach 100. However, the most notable one is this species, the Wallostemenius. So, Nava'i Kolu has found that there's a ratio of the 40,000 invasive species to one native species, and it just so happened that they found one native species on that day. However, something else more incredible is that the eDNA method also found that one species on that day, showing that this method can be used to detect native as well as invasive species. And this is just a quick comparison between the two samples. You can see initially much better non-fish DNA results, and also much more yellow and much more reads. So although my data doesn't match entirely with Nava'i Kolu's, I believe it provides an additional method that can be used by Nava'i Kolu. If anything, my data proves that, as you can see, that Stereovics 1, 2, and 3, so that was the same water collection sample, same time, same day, same location, but had varying results. So I know that this proves that if eDNA method is going to be used, it's likely going to need lots of trials from the same place in order to counteract things that others have missed. I believe that eDNA can be a successful method, however, there needs much more refining. If only through 2 runs I was able to have huge improvements, it's likely that more trials and more errors will lead to much more benefits as well. For example, I could learn about whether centrifuge or stereovics would have a better results, or even if there's a way to mitigate the errors such as species can be used as the genera. However, as it stands now, there is still much that needs to be done, but this makes sense as eDNA is a relatively new method, and new technologies and new procedures are being tested every day. I have no doubt that in the future, eDNA will be a very successful method to use by Nava'i Kolu. And so a reflection of what I learned is that research and experimenting takes a lot of time and patience. I thought that I could go out there and figure it out really fast and get my research and my data, but that was not the case. And secondly, I learned to be okay with not knowing everything. Dr. Chan told me that this is the test that I took a whole college course on, and it's not something that I can really learn in just a few days and few weeks of reading papers. Some advice that I had was definitely to find something you're passionate about. In the time and the hours I mentioned previously, it went by a lot more enjoyably and more fun when you have something that you really care about and you really enjoy. And lastly, so don't be afraid to ask questions. You're not going to know everything, and you're not going to know it by just being quiet. So be bold, be brave, and just ask questions to teachers and mentors. Lastly, I want to say thank you to Dr. Chan for helping me through the entire project, showing me the way the whole time. Ms. Kobayashi for actually going out into the streams with me and collecting water samples. Mr. Tong for teaching me about the DNA sequencing and how it works. And lastly, Dr. Boric for allowing me to use his room for DNA extraction. And lastly, to all of you for listening to my project. Hope you have a great day. Thank you very much.